Hello students. In the previous lectures we have discussed about the pathways, that is EMP, HMP, ED and pentose phosphate pathway and hexose phosphoketolase pathway. Pentose phosphoketolase pathway and hexose phosphoketolase pathway. That was the end of the second chapter. I will start with the third chapter of microbial metabolism, and in that chapter we have to discuss about the aerobic pathway for metabolism, in which we will be discussing about Krebs cycle specifically and respiratory electron transport chain. Pyruvic acid at the end is the end product of glycolysis, that is EMP pathway, that does not enter citric acid cycle or TCA cycle directly. The 3 carbon atom molecule of pyruvic acid is first changed into 2 carbon atom acetic acid molecule. The 1 carbon is released as a carbon dioxide. The acetic acid, on entering in the mitochondria, unites with coenzyme A to form a acetyl coenzyme A, active acetate. This reaction is catalyzed by several enzymes collectively known as pyruvate dehydrogenases. The two hydrogen atoms are released in this reaction and accepted by NAD to form NADH plus H plus ion. The pair of hydrogen is passed down to electron transport chain system, and 3 ATP molecules are generated in this process. Since 2 pyruvic acid molecules are formed during metabolism, from one molecule of glucose, 6 ATP molecules will be formed during the oxidation of 2 pyruvic acid molecules. This is the first step in the pyruvic acid oxidation, or we can say citric acid cycle or TCA cycle, Krebs cycle. It is also known as Krebs cycle. Pyruvic acid is converted into ethyl alcohol if in the anaerobic condition or alcoholic fermentation, or lactic acid, that is lactic acid fermentation. In the absence of oxygen, anaerobic condition, that is in the anaerobic condition. In the presence of oxygen or in the aerobic condition, however, the metabolism is different and pyruvate is oxidized to acetyl-CoA, which then undergoes a series of changes referred to as Krebs cycle or Krebs citric acid cycle. The citric acid cycle is known as tricarboxylic acid cycle also, since citric acid is a tricarboxylic acid, that is 3 COOH groups are present in this citric acid. Each cycle result in the conversion of one molecule of acetic acid into acetyl-CoA to form a carbon dioxide and a water molecule. Then the cycle, actually this cycle is like this, where pyruvate gets converted into acetyl-CoA. Here 2 hydrogen molecules and one carbon molecule is released, which the carbon molecule released is as a carbon dioxide, and the hydrogen molecules released get captured by NAD plus ions to convert it into NADH plus H plus ion, that is in the electron transport chain, which gives you 3 ATP molecules. As we are dealing with the 2 pyruvate molecules, we will get 6 ATP molecules. The acetyl-CoA then on reacting with in the presence of citrate synthetase enzyme, the acetyl-CoA reacts with oxaloacetate to give you a 6 carbon atom, that is citrate. The citrate molecule then dehydration reaction occurs with the citrate molecule to form cis-aconitate with the enzyme aconitase. Aconitase enzyme again reacts with cis-aconitase to give you isocitrate molecule with the hydration of cis-aconitase. Here is a dehydration-hydration reaction run by aconitase enzyme to give you isocitrate. This isocitrate then releases two hydrogen molecules to give you 6 ATPs on a dehydrogenation reaction with isocitrate dehydrogenase, resulting in oxalosuccinate. The oxalosuccinate then releases one carbon as a carbon dioxide and converts into alpha ketoglutarate in the presence of carboxylase enzyme. And the alpha ketoglutarate, that is 5 carbon molecule, dehydrogenates to succinyl coenzyme A. As coenzyme A is added in this and succinyl coenzyme E is produced, alpha ketoglutarate dehydrogenase is the enzyme. This is the, and here also one carbon dioxide is released and 6 ATP molecules are produced with the release of 2 hydrogen atoms. Then succinyl CoA, that is 4 carbon atom containing succinyl CoA, is converted into succinate with the release of coenzyme A, and this is the phosphorylation of ADP. Succinate thiokinase is the enzyme working on it. Here GDP molecules are converted into GTP molecules. Succinate then converts into fumarate in the presence of succinate dehydrogenase. Here release of 2 hydrogen atoms occurs, and here FAD is converted into FADH2 in the hydrogen transport system, releasing 4 ATP molecules. The fumarate, 4 carbon atom fumarate, is converted into malate with the hydration reaction. Here water molecule is added to give you a malate molecule. The 4 carbon malate then converts into oxaloacetate, releasing 2 hydrogen molecules in the NAD to NADH2, giving you 6 ATP molecules. Here dehydrogenation occurs with the malate dehydrogenase enzyme. This is a cyclic reaction. Then oxaloacetate again reacts with acetyl CoA to give you a citrate. The cycle is completed. In this Krebs cycle, actually we have a number of reactions and that passing into electron transport system to release ATP molecules. There are actually 6 ATP molecules in the first step, that is pyruvate to acetyl CoA. Here 6 ATP molecules are produced. Then again in the fourth step, dehydrogenation reaction with isocitrate dehydrogenase, here also 6 ATP molecules were produced. In the sixth step of dehydrogenation reaction, here also we have a 6 ATP molecule generation. In the seventh step, that is phosphorylation of ADP, succinate thiokinase action, here GTP is produced, which is equivalent to ATP if you observe, and 2 GTP molecules were produced. In the eighth reaction, that is dehydrogenation reaction, again we have FAD converted into FADH2. Here 2 FADH2 were produced and giving you a 4 ATP molecules. Then in the 10th reaction, the dehydrogenation of malate to oxaloacetate, NAD is converted into NADH2, giving you 6 ATP molecules. Actually, in these reactions, total of 30 ATP were produced. If consider the first reaction as pyruvate to acetyl coenzyme A. And in the citric acid cycle or Krebs cycle stage 3, we have 30 ATP generation. Gross ATP generation is 30 ATP. In this, all the reactions occur and give you electron transport, passes into electron transport system. Actually, all the ATPs are produced during electron transport system, and NAD/NADH2 transformation or FAD/FADH2 transformation occurs here, giving you ATP generation. This is the citric acid cycle or tricarboxylic acid cycle or Krebs cycle. Also, oxidative phosphorylation or oxidative metabolism of pyruvate or pyruvate oxidation occurs in this Krebs cycle. Here, important steps are the first step, that is pyruvate to acetyl CoA. The second important step, that is production of ATP, if you observe, is isocitrate to oxalosuccinate. Here 6 ATP molecules produced. Pyruvate to acetyl CoA, 6 ATP molecules produced. Then alpha ketoglutarate to succinyl CoA, 6 ATP molecules produced. Interestingly, succinyl CoA to succinate, here GTP is produced, which is equivalent to ATP, giving you 2 ATP molecules. Then succinate to fumarate, FADH2, here 4 ATP molecules produced, and malate to oxaloacetate, here 6 ATP molecules were produced. In total, here 30 ATP molecules were produced, and complete metabolism of glucose gives you around from EMP pathway to Krebs cycle or through RETC chain, it is 38 ATP gross ATP generation was there, or net ATP generation was 38 ATP molecules. So this is the Krebs cycle or citric acid cycle. We will discuss respiratory electron transport chain in the next lecture. Thank you very much.